All right, welcome everyone. If you haven't signed in, go ahead and sign in for us so we know who all is here. Want to extend a very special thank you to everyone at HSC for helping make this possible. We're really excited, especially being able to work with them. And here to introduce, we have Dr. Rance Berg, has dedicated over 30 years to studying molecular and cellular mechanisms that regulate innate and adaptive immune responses. After earning a BS in biology and a minor in chemistry from DePaul University, Dr. Berg has worked as a research technician before completing a PhD in immunology from University of Colorado Health Science Center, with research for the National Jewish Medical Research Center, focusing on T cell development and functions. As a postdoctoral fellow at UT Southwestern Medical Center, Dr. Berg made groundbreaking discoveries on how memory CD8 plus T cells contribute to innate immune responses. Currently, he is a faculty member with the University of North Texas Health Science Center. His research centers on understanding immune responses to infectious and inflammatory challenges, particularly the role of T cells, NK cells, neutrophiles, and monocytals.
That's pretty close.
A dedicated mentor, Dr. Berg has guided numerous PhDs and MS students, technicians, undergraduate students, and has done all of that while serving on over 75 graduate thesis and dissertation committees. He currently teaches immunology and holds key administrative roles, including the Assistant Dean of Graduate Education and Admissions and the Director of MS PhD programs. He is also the chair of the Institutional Biosafety Committees and still has time to review journals for both NIH and VA. So with all of that, thank you so very much. Come on in and tell us about yourself.
Awesome. Thank you so much, Ian. Thank you guys so much for being here. What a very generous introduction. The reason I'm able to do all those things is because I collaborate and partner with a lot of people, including Miss Autumn Young. She gets these things set up. I don't reach out and make all these things happen. And the reason I could be so productive in my lab is that I had young scientists like you guys in there working for me and helping me. So this is gonna be kind of a different type of talk. I decided not to go like hardcore immunology. So by show of hands, which of you guys are freshmen? Okay, sophomore two, juniors, seniors. Okay, so we have a lot of seniors. You guys are right at that precipice of your next step, right? What do you want to do? I'm just here to talk about what I did and what worked out for me. Maybe a few things that didn't work out for me, but ultimately it's up to you guys to find your career path. And what you'll see me emphasizing is take advantage of opportunities, partner with people, and put yourself out there. And sometimes that can be a little bit scary, right? But the more you put yourself out there and interact with people and take opportunities that are presented to you, the more options you have for your career. Next slide, please, Ian. Is it picking me up on the mic or?
Yeah, can everybody hear?
Do I need to mic something up?
I think you're good. Yep, you're good. Yeah, if you just want to take like one more step this way, perfect. There you go.
Awesome. Is that good?
Yeah.
Okay, next slide. So,
Oh, I thought that was the next slide.
there we go. That's the next slide. That's where I want to be. I was born outside of St. Louis, Missouri. There's my father there and me and my brother, and we ran around in the woods a lot. We fished and we caught things, and quite honestly, we cut some things up and looked at them, and we were both just very interested in what was going on around us. Fast forward 10 years, next slide, and my dad said, hey, I'm gonna start a business in Amarillo, Texas. And I was like, oh my gosh, I'm going from the woods of Missouri to this big flat open area in Texas. But I met a lot of people there, and I really got into playing soccer, and I thought, hey, maybe I'm gonna be a professional soccer player, right? As we grow up, our career aspirations change. And I really thought, you know what? I am pretty darn good. Fast forward, I go to school at DePaul University on the next slide. And I started playing soccer there. And then I figured out, well, maybe I'm not as good of a soccer player as I thought. There's a lot of other really good soccer players out there. But I really enjoyed my time there. And I got introduced to one of my biology professors and was given an opportunity to work in the lab. The first thing I did in the lab, I cleaned test tubes and I autoclaved things. That's what I did. That was my job. All right. Didn't hardly pay anything, but I thought, well, at least I'm in a lab. This is kind of cool. I think I might want to be a scientist or something. Dr. Sidney Beck gave me the opportunity to work on a research project, and it really kind of inspired me to think about, you know what, I really do like this. I wasn't sure what I wanted to do. So after graduating, fast forward, I went back to Amarillo, Texas, and I get a job working in the Harrington Cancer Center. And my mentor there, Dr. Carl Pierce, he was studying the role of a particular cell type and how it could suppress immune responses. And we're going to talk a little bit more about immunology in a couple of slides, but I want to give him a specific shout out. He had organized a conference up in Montana, an immunology conference, and he said, hey, do you want to go? And I said, I'd love to go. I can't afford it. I just got married. I had young kid there. It's my dad, my mom. It's like, I can't afford to do this. He said, you know what? I'll pay for it. He said, do you want to go? And I said, well, heck yeah, let's go. I go up to this conference in Montana, meet a bunch of people, including somebody who's in graduate school in Denver. And I was like, ooh, I like the mountains. This sounds pretty cool. Maybe I'll apply. Fast forward. I apply to several schools and I get into the PhD program at the University of Colorado Health Science Center. And I started working with a German gentleman by the name of Dr. Uwe Sterz. He had an MD and a PhD. He was really brilliant. You know what else he's really good at? Flipping houses. He'd show up to work and he'd have paint in his hair. Right. So he was one of these people that did all these different things. And I'm like, oh, that's cool. Like, I don't have to just spend 20 hours a day working in the lab. There's other things I can do. Next slide, and I'll talk about a few things I did. So I really got into immunology as a technician. I never took immunology as an undergrad, but as a technician, and then get into this top-ranked immunology program, I was like, ooh, this is a little bit scary. I've never even taken immunology, but I learned a lot on my own. I figured out what I didn't know, and that's an important lesson. You've got to know what you don't know. Then you can kind of work on figuring it out. What does the immune system do? Well, it has to recognize things, and it has to be able to distinguish self from non-self, right? If you think about what our immune system is doing, it's trying to fight off all the bad things. But what we don't want it to do is get confused and attack ourselves, right? It performs different effector functions. And this is related to T cell development and B cell development, right? But these are the acts of the neutrophils, for example, gobbling up bacteria and killing them. The T cells poking hole in cancerous cells. Immune regulation. The immune system has to know when it's time to stop. Right? It doesn't always work so well because sometimes when there's a lot of allergens, like right now, our immune system kind of keeps going and going. We have to sort of figure that out. And then importantly, the immune system shows memory. And I say vaccines, anyone. The basis essentially of all vaccines is the development of T cell and B cell memory, such that when you're exposed to that antigen or that pathogen, again, we'll use SARS-CoV-2 as an example, your immune system real quickly says, oh, I remember that. Right? It has this ability to say, I've seen this before. I need to do something. I need to do it quick. And we have innate immunity, which provides very early immune responses, and then adaptive immunity mediated by the T cells and B cells that occurs later. Next slide. I'm going to show you guys quite a bit of data, but I'm going to hit the high points of it. Okay. So what we did is we were interested in understanding how T cells developed. So I used molecular biology techniques, and hopefully you're learning some of these things in the lab, to clone out a specific T cell receptor from a set of T cells that could recognize Listeria monocytogenes. If you haven't heard of Listeria, it's the culprit in Bluebell ice cream. All those years ago, they pulled every Bluebell carton off the containers and people went crazy. Contaminated with Listeria. Listeria can cause anything ranging from very mild gastroenteritis to even almost no symptoms, all the way up to systemic infection, spontaneous abortion, central nervous system infection, septicemia, and even death, particularly in immunocompromised people. So we're interested in understanding how we generate T cells. So we made this TCR transgenic mouse. It's just a mouse that all the T cells are exactly the same. Now, in the real world, that's not what we want. We want to have a bunch of different T cells, but this made it easier to study. And we bred it onto these different mouse, different backgrounds. And what you'll see here in these upper three, A, B, and C, is the presence of the T cells that are specific for Listeria. Down here, in some strains of mice, we couldn't generate those T cells. So next slide. So what we did is then we utilized the ability to make what are called fetal thymic organ cultures. We take fetal pups from mice, we pull out their little thymic lobes. The thymus is where the T cells develop. We can put them in a dish and we can add back things and ask, now can we induce the development of the T cells? And when we add it back in a particular peptide that comes from the mitochondria, actually, because it has a formal group on it, we're able to generate the T cells as shown in A, B, C, D, and E being controls down there. And those T cells were functional. So we figured out how those T cells developed. It's critical for us to understand how the immune system develops so that if we need more of something, we can help make more of something. And if we need less of something, we can make less. Next slide. So we published several nice papers. Next slide. And then we took this cool picture. There's my boss right there in the middle, Dr. Uwe Stertz. He'd show up to work with flip-flops, shorts, and Hawaiian shirts. And he was is still very eccentric and very smart, but he gave me the freedom to do whatever. So I say, what did I learn there? Work hard, play hard. Take your kids with you. Okay, you already heard, I chair the biosafety committee. We can't take our kids to work anymore. Back in the day, I was divorced then. When it was time to pick up my son, if the experiment wasn't done, I went and picked him up, bring him into the lab. I bring him into the lab one day and I have my ice bucket sitting there and it has all of my little mouse pups that I just harvested from a pregnant mouse, ready for me to dissect them out. And I bring him into the lab and he's looking up there. I'm gonna pick him up so he can see it, right? And he looks down there and he goes, whoa. I said, what is it, son? He said, babies. I said, you're right. Those are babies. And then the next question, right? The first question was easy. What kind of babies are those? And then his eyes got huge and he turns to me and he goes, humans? And I said, no, son, I don't have a bunch of little human babies in there. They're from mice. He was fascinated. He was like, this is the coolest thing. Another time he stuck a juniper berry up his nose. And I thought the only way I'm gonna be able to get this out is with some really cool forceps I have at work. The weekend, I bring him up, lay him out on the lab bench. And I'm like, okay, you better hold still because if you jerk when I do this, the juniper berry is gonna get in your brain. You know, you go, oh my gosh, got the juniper berry out. It was good. Perform good experiments. What does that mean? A good experiment. Sometimes you don't have to perform the perfect experiment. You just need to perform the experiment and have the appropriate controls. My boss taught me that. Perform unknown experiments. A very famous immunologist, Dr. Merrick. I was going up to run an experiment one day and she said, she was British. I won't use the British accent because I've been told it's not very good. But she said, well, what are you working on? And I told her, oh, it's a control experiment. I know what's going to happen. And she looks at me and says, why would you do an experiment if you knew the outcome, right? Obviously, she was being a bit facetious because sometimes we have to do control experiments. You know what's going to happen. You just have to do it, right? But her point was do experiments where you really have no idea what's going to happen. Of course, we always want to have a hypothesis, but that's an important lesson to learn. And sometimes you say, I have no idea what happened when I add these 3 chemicals into this dish, right, with these cells. I have no idea, but I'm going to try it. Meet with students, Dr. John Kapler, super famous guy, Howard Hughes, National Academy member, and he was on my committee. And I go see him sometimes, and he'd be in the lab still working. And I'd ask him, hey, is it okay if we talk? And he'd put up a finger, he'd be like, busy now. And I knew, okay, right? He would come find me, right? And I think that that's really important. He showed to me that it doesn't matter what level you're at. Meeting with students, answering their questions, working with them is incredibly important. Treat your lab like family and don't give up, right? PhD is hard, and MD is hard, PA is hard, PT, any of these degrees that we offer, they are hard. Don't give up. Next. So back to Texas it was. I went back to UT Southwestern, got the opportunity to work with Dr. Jim Foreman and decided, I think now really I'm an immunologist, like I can do this. So what projects did I embark on? Next slide, please. This is a control experiment. You don't need to know all the details, but basically we had mice that were infected and then we exposed them to macrophages and we saw that the T cells could respond, okay? And that was expected, right? But up on top, naive means those mice had never seen Listeria before, right? Generally, if you haven't seen a pathogen, your immune system, the first time you're exposed to it, you don't get a very good response, okay? That's why oftentimes with vaccines, we do boosters to boost the immunity. And so this was a control up here, and I saw that data, and I said, we got something going on there, boss. And he said, that shouldn't look like that. Your T cells shouldn't be responding like that. And I said, but they are. He said, do it again, did it again, did it again, right? We took a simple control experiment and we parlayed that into probably 10 or 12 publications and probably $3 or $4 million worth of grant funding off that experiment right there. It's a very, very simple experiment. Next slide. So then we're like, okay, well, we did the simple experiment and we saw something that's kind of interesting, but we have no idea how it works. So let's begin to sort it out. And so we used mice, once again, a TCR transgenic, and we transferred T cells into another strain of mice where we could track them. So then we could follow those specific T cells. And then we would prime them with different pathogens, vaccinia virus or vesicular stomatitis virus that had an antigen, so we could track these T cells. And then after either 7 days, which would constitute an effector T cell response, or greater than 4 weeks, that'd be a memory response because the pathogen is cleared, but you still have T cells, right? Next slide. And what did we see? Interestingly, what we saw is that if you took, and I think this was memory T cells, and you exposed them to interleukin 12 and interleukin 18, these are cytokines, they're small proteins that are soluble. When they bind to the receptors, they can have specific effects. What we saw is these memory T cells could respond to those cytokines in the complete absence of an antigen. And what that means is they're kind of non-specific in nature and they can respond very quickly. It's sort of like a stopgap. While you wait for your T cells to develop, you can have other T cells that can respond very quickly. So this is all good and fine. These are a bunch of other controls to show it was definitely mediated through 12 and 18, that when we block those antibodies, our response goes down to nothing. But at that point in time, this is, how do I call it? We call it like phenomenology, right? We've done something, we've seen something, there's a phenomenon. I have no idea if it's important or not, right? Next slide. Kind of a convoluted way here to now make these T cells specific for one thing and then transfer them into mice that are very immunocompromised because they can't make this certain cytokine called interferon gamma, and then ask if these T cells can provide protection against the subsequent infection. Next slide, please. So indeed, when we infected these mice, if you look at the immunodeficient mice, what's shown here is the number of colony forming units, right? If we take bacteria that are isolated from somewhere and we put them on an agar plate, we can count the number of bacteria that are present. And then through some relatively simple math, we can back calculate how many bacteria are in the spleen and the liver, which are the target organs for Listeria. So here's our control mice. They have very low bacterial counts. Here's our immunodeficient mice. They have very high. And you see that when we provided back these memory T cells that were not specific for this pathogen, the CFUs go down. So we're able to show that there's actually a function for these T cells, right? And then you can start thinking about therapeutics, right? Start thinking about, hey, if somebody is infected with something, or if in the future we figure they're going to get infected, SARS-CoV-2 again, right? Is there a way that we can boost their immune system in addition to vaccines so that they can respond more effectively and efficiently? Next slide. This is just showing that the T cells perform better than the natural killer cells by providing more protection. Next slide. And so we published several articles, including we had the cover of Journal of Immunology, as we were doing immunohistochemistry, and we were tracking where these T cells were located. And what we're able to show is that these T cells, they co-localized in the areas in the spleen and liver where the Listeria were present. So where the bacteria were, that's where the T cells went. Right? It's important for our immune system to be able to home to those technician, I don't have, yeah, oh yeah, technician. Well, those guys were technicians. I was a postdoc, and they worked very closely with me. MD, PhD, works for Luminex, a big company, doing all sorts of good stuff. And Sean is still a technician, a very, very good technician, right? So they came in with their bachelor's degrees and they said, I want to do something. I want to make a difference. And they were smart and they were willing to work hard. They got publications. They went on to do really, really good things. Next. All right. Let the data guide you, right? We want to have hypothesis. You know, most research should be either hypothesis driven or exploratory in nature. But if it's hypothesis driven, then you want to say, we hypothesize that when we have these cytokines, this specific thing will happen through this particular mechanism. I would guess that about 50% of my hypotheses are wrong, right? That's okay, right? You thought something would happen, but then you do the experiment and you're like, that's not exactly what happened, right? You must let that data guide you. I'm gonna go back to taking advantage of opportunities. I was able to be involved in writing animal protocols, biosafety protocols. I served on search committees. This is as a postdoc. I wasn't a faculty. I wrote grants. I helped manage the lab. My mentor was very generous with that. He let me hire technicians. They worked for me, right? So I got to learn how to mentor and how to guide people. I attended a lot of meetings. I ordered and I did stocking and I took inventory and I think, oh, well, you know, probably just had your technician could do it. When I started my lab almost 20 years ago, there was zero in that lab that I didn't know how to do because I've already done it, right? And so don't ever think anything is beneath you or that, oh, somebody else can do it, right? Oh, that's not my problem. I don't need to do that. Take advantage of things, right? Explore diverse research projects. As I said, I think it's important to reward not just our technicians, but everybody around us. Anybody that helped me with my manuscripts, I offered them authorship on the paper. It didn't matter if they had a bachelor's degree, no bachelor's degree, a PhD, an MD, it didn't matter, right? They contribute to that project. Work hard, play hard. Same things, right? You got to enjoy life, right? Work-life balance is important. Next. I had high impact publications. I had a postdoctoral fellowship. I have a K award. I had ideas for projects. I thought I could manage a lab. I had plenty of confidence. I'm like, all right, I'm going to apply for faculty positions because that's what I wanted to do. Many people in my shoes, maybe they would have went into industry, or maybe they went into, you know, more of a teaching position or whatever it happens to be. I wanted to run a lab, but I also wanted to teach and mentor. Fast forward, next slide. And I just went right down the road from UT Southwestern, right up there in Dallas, and just drove right down I-30. I'm like, oh, cool. I landed at another institution that's good and has projects. I was divorced, but my ex-wife and my son still lived there. They lived in Fort Worth, and so it made it even easier for me. I wanted to be close to my family. And I know that's important to a lot of people. So even though lots of times I would shoot out and went to Chicago and went to Denver, lots of times I kind of ended up being back around my family because that was important, you know? So I'm an immunologist, but can I run a lab? Can I teach at the graduate level? Can I teach aspiring physicians? Do I have the ability to serve on committees? I don't know. Next slide. I set up my lab, and I was so fortunate to recruit Karen Carr. She was a graduate from Dallas Baptist University. It's right down the street. Josh Balch, he was a technician in my lab. He graduated from Midwestern State up in Wichita Falls. And then Dr. Amy Sieve, she was a postdoc in my lab. Within 6 months of setting up my lab, I had these guys in there. I would have failed miserably without them, right? People are your most valuable assets, right? We're all smart, we can work hard, but partnering with people and working with people, recognizing what they can do compared to what you can do, and working with people's strengths is so important. Next. I'll talk about a little bit more data, some of the data that I generated in my own lab before I finish up. So I want to give Autumn a couple of minutes to talk about some things. Interleukin 12 was a cytokine that we'd studied. As I said, cytokines, small proteins, bind to receptors, they're soluble, and they influence the cells that they bind to. There was a brand new cytokine called IL-23, and it was in the same family as interleukin 12. And so we had the not so brilliant idea of, well, let's just see if we get animals that lack IL-23, KO means knockout. It's just a genetically engineered mouse that cannot make that cytokine. Let's just see how they do when we infect them with our go-to pathogen, which is Listeria monocytogenes. This is a survival curve. If you look at the black circles up on the top, what that's showing is after infection, over the course of 12 days, essentially all of the animals were still alive. I think we lost like 1 out of 10 or something, 1 out of 11, I don't remember. But what you see within 10 days, all the animals that lacked interleukin 23, they all died. Pretty straightforward experiment to interpret, right? You need IL-23 to be able to resist bacterial infection with Listeria. Shown over here is just the CFUs, once again, the colony forming units, so the amount of bacteria that could be found in the spleen and liver at 2 different time points. And you see both times there's more bacteria. So those animals couldn't control the infection. This is phenomenology, right? At that point in time, it's like, oh, that's super cool, right? We got a good result, but we don't know why. Next slide. We knew that IL-23 could help regulate the production of another cytokine called IL-17A. It was brand new on the scene as well. And what's shown here, if we just focus on the white versus the red bars, that if you looked in the wild type B6 mice compared to the mice that lacked interleukin 23, the amount of IL-17A in the spleen and liver was reduced over different types of conditions. So IL-23 regulates IL-17, right? So then the next logical step, try to think pretty linear. Some of my students think a little bit tangential and circular, and that can be okay. I try to think pretty linear. If IL-23 mice die and IL-23 regulates IL-17, one might hypothesis that IL-17 deficient mice wouldn't do well either. Next slide. We were able to get mice that lacked the receptor for IL-17, and it phenocopies, right? You get the same result in those mice that lack the IL-17 receptor as we did in the mice that lacked IL-23. So these 2 cytokines are both involved, and you can see the increase in the bacterial burden in those mice that lack the receptor for IL-17. Next slide. Importantly, when we now did a rescue type experiment, right? If we know the IL-23 knockout mice don't do well with bacterial infection and it regulates IL-17, what if we just inject back? So this is a therapeutic, right? we can inject back that IL-17A, and what we see compared to the IL-23 knockouts is that the bacterial burden goes down. There's less bacteria in those mice, so that IL-17 is able to help rescue those mice. So, once again, although my lab has never studied like therapies for infections or cancer, we're always thinking about it. We're always thinking about, well, could this contribute to a vaccine? What could this do? Next slide. Finally, what we're able to show is that what was being regulated by these two cytokines was the recruitment of neutrophils to the liver. Okay. And so if you look at different hours post-infection with Listeria and then look at the percentage of neutrophils, here's our wild type animals, and then the mice that lack IL-23 and lack the IL-17 receptor, they have fewer neutrophils, right? And so that led us down this path once again of saying, okay, 23 leads to 17, leads to neutrophils. What are the neutrophils doing, right? If there's reduced neutrophils and the mice are sick, maybe it has something to do with them. Next slide. So we had published that. So we used to go on retreats. This is one of my good friends and colleagues. Some of y'all maybe have met him. Probably Ian has. His name is Dr. Harlan Jones. He's one of our faculty members. And we're playing volleyball, right? And he had just dove, right? We're at this retreat, having a great time up at Lake Texoma. And I'd like to think that I was cheering for him. I'm not sure exactly, but someone took that picture and we had it outside of our lab forever. And Dr. Jones and I, we had a good time laughing about it. Next. So we went back to the neutrophil story, and I'll just talk about a couple more pieces of data here. And what we're able to show is that when you give a high dose of infection with Listeria and you used an antibody to deplete the neutrophils, okay? So you inject this particular substance and it removes all the neutrophils, right? So now these mice don't have neutrophils circulating. What we were able to show is that those mice succumb very rapidly to infection with Listeria. So you remove the neutrophils, the mice all die. So neutrophils are important for controlling the infection. Next slide. And I'm not going to talk about all this crazy data, but one of my more recent students who graduated a couple of years ago, she was interested in, well, if neutrophils and monocytes, and I skipped that part of the story, are both involved, what do they do differently, right? These are both innate immune cells that can phagocytose or eat the bacteria. And she got a reporter strain of bacteria up there. It's called Act-A LMGFP. If we just break all of that down, it's a strain of Listeria that expresses green fluorescence protein, but only when the bacteria escape into the cytoplasm of whatever cell has engulfed them, okay? It took her a lot of time to just set this system up where we were infecting these different cell types and asking questions about, well, do the cells eat the bacteria? Do the bacteria get in the cytoplasm? Do the bacteria blow the cell up and escape? Next slide. Ultimately, what we were able to show through what we termed as a containment index, so we kind of coined that. We kind of came up with, okay, if you take up the bacteria, if you contain it into a phagocytic vesicle where then the bacteria can be killed, you've contained the bacteria. If the bacteria escape into the cytoplasm, then they replicate and they can explode the cell and go infect other cells. So what we're able to show in the bone marrow, liver, and spleen, the neutrophils compared to the monocytes had a higher containment index. They were good at taking up the bacteria and holding them and ultimately killing them. We did some other experiments to further prove that and then published that a couple of years ago. Here's some of the guys in the lab. This is Naomi. And she was maybe a little bit clumsy. And so at some point in time, we had all the bubble wrap. They decided to wrap her in the bubble wrap. That way, if she fell down, she wouldn't get hurt, right? Both those girls were originally from Nigeria. And so they're all into this whole concept of, remember the Jamaican bobsledders, right? From Jamaica. And so at some point in time, we were moving labs and I had no idea what they were doing there. Next slide. So basically, we came up with a working hypothesis. Interleukin 12 does this, interleukin 23 does this, and we put together this nice figure. We published some review articles, and all was well. Next. So several other publications, other people in the lab. Stephen was always sleeping. He was a master's student. He got his MD from Texas Tech, and he's a practicing physician. I hope he doesn't still sleep when he's at work, but he was so wonderful. Amy was another master's student that published a paper and then went on to do her PhD in Montana. And then there's a couple of my early postdoc, who is a tenured faculty member at Tarrant County College, Northwest Campus, and Karen, who is a global medical director for AbbVie. AbbVie is a huge pharmaceutical company. She's in Spain right now. The other day she was in Sweden. She's all over the place. Next. We're going to skip this next set of data. So skip. So that first figure earlier that had all the color and the pretty things on it, Karen made that. My student Tim made this one. I was like, Tim, that's not. See, I say, how does ECSOD function during bacterial infection? This is just an enzyme that we're working with, right? Or how not to make a conclusion figure. I'm like, dude, we can't, this is not gonna work. Next. And published several more papers. There's Tim. Tim's wife had a baby when Tim was a PhD student, and he was incredibly supportive of his family, but he kept working too. Anytime he could get, man, he'd come up to the lab, he'd work at nights and he'd do these, different things. He was just very dedicated and a great family man. And he is a senior research director at AstraZeneca. So another big company, and is just really doing well. That's Mohana up there. I kind of lost track of her for a while. She went and did a postdoc with one of my friends up at Northwestern University, and then went out to California. And I kind of lost track of her for a while, and I finally tracked her down. I said, "What are you doing?" She said, "Oh, I started a company." So she spun out a startup company with some things she was working on, and my fingers are crossed she's going to do well. And that's Allie, and she went and did a postdoc at University of Virginia, and now she's a senior research scientist at a startup company called Arcelix. Next. So what do I do now? I don't do as much research, but I work on recruitment, admissions. I work on our teaching assistantship program to try and provide all of our students good opportunities, because if you can't teach, you're not going to do well running a lab, and you're certainly not going to do particularly well in academia. We really have transformed our graduate education into a model that's competency-based and individualized. I can't tell you guys what you might need to get ahead, right? I don't know what you want to do, so I would work with you. And then importantly, create diverse career opportunities for graduates. Of all my students have graduated, none of them are in academia. My postdoc, she's a faculty member at Tarrant County College, but we know people do a lot of different things. Study something that you find to be interesting and important. Don't let anybody else define that for you, right? And then apply for jobs and pursue a career that you believe you will enjoy. Not your parents, not your significant other, not, the newspaper, that you think you will enjoy and that you can make a difference. Next. Acknowledgements. I pretty much hit all those people. Here's a bunch of my collaborators. Next. Other acknowledgements, of course, I want to mention Autumn again and other people that I've worked with. Next. I'll just try to end on, so my parents ended up moving up to New Mexico. And so we still go up there. There's my father. He passed away about 5 years ago. There's my son. He turned into a computer programmer. So he went to UT, and I thought he's going to be a biologist. And nah, he's like, Dad, I'm good at programming, and I can make good money doing it. So, okay, then that's what you're going to do. So he programs. Just a few other pictures of different people. And which one do I want to end on next? Let's just end on that one right there. There's my wife and my 2 stepkids. There's my son and his wife. And whenever I can, I try to go fishing. I showed you the first picture of me standing next to my dad in Missouri holding a little fish, right? I've kind of stepped it up now. We go fishing in a lot of different places. That was probably a 250-pound blue marlin that I caught 10 years ago or something like that. So I'm going to end on that so that Autumn has a couple of minutes to talk. I know that was a lot. I don't expect y'all to like have deep diving scientific questions, but if you do, or you have any questions about anything, just let Autumn and I know, okay? Thank you so much.
Thank you so much. That will interject for just a second.
Oh, yeah.
Please, everyone here, please complete, and for those of you online, QR code works as well. This is the Bridges Across Texas-LSAMP Alliance event survey. Please complete it. It helps us be able to do more things like this.
Yeah, thank you so much for the generous offer for us to come out here and do this. We really love to do it.
Thank you so much. And Autumn.
Yeah, I think there were a couple of you who snuck in that didn't get one of these brochures, so I just wanted to make sure. Can you raise your hand for me? Amazing. Good to see you. Awesome. I also have a QR code for you. I'm just gonna leave it up here for you guys to scan. If you want to get on our email list or anything like that, if you scan this, let us know what program you're interested in. You'll start getting those like marketing emails. We don't bombard you, or at least I don't think we do. And then I also have a few business cards. If you want to reach out to me directly, I can give you one of my cards. I'm the primary advisor for graduate programs in the College of Biomedical and Translational Sciences. So PhD program, master's programs. I also was the main advisor for the medical school TCOM. So if you're interested in pre-med or anything like that, I can also definitely help you navigate that process as well. And then Ian, if it's okay,
Yes.
can I jump on the computer?
Absolutely.
I'll reshare the screen so that way the people online could
Perfect. There you go.
go on. I just wanted to see, or let's see.
And then I'll just drag it over.
Okay, sweet. I just wanted to show you guys how to access advising, so that way you can stay in touch with us. You said just drag it over here.
Yep. There you go.
Amazing. Okay, so obviously you know how to go to our website, right? So when you get to the main page. Okay, if you go to apply now.
There's one of our PhD students right there.
Oh, yeah.
Sorry.
That's okay.
Shameless plug. All our programs are good.
Yes. If you go to apply now, and then if you click on schedule advising down here, you can also look at our calendar for any sort of upcoming events that we have, which we have a really big one coming up that I wanted to plug on December 14th. It's a Saturday. So yeah, go ahead and put that in your calendars. Also, if you get on our email list, we will send you some emails. When the tickets go on sale, they should be going, well, sale, they're free, but they should be available for you to reserve within the next week or so. They'll be by program. But essentially it's like our giant open house. So all of our programs are going to be available. Dr. Berg will be there. We do some faculty networking. We have current students. And then usually we have the deans present for most of our programs as well. So I just want to make sure that y'all have that information. It's a really fantastic opportunity to get to know people and explore other options. We also have some fun stuff to do as well. Okay, if you click on schedule advising down here at the bottom, and I should be right at the top. So right here, this is me. I just cut my hair. I got married and I was like, I needed to change. So I have really long hair in this, but scroll down just a little bit for me, Ian. Perfect. This is my contact information. Again, these are the main programs I advise for, but I can really answer questions about any of our programs. So clinical-based programs, any of the undergraduate degrees. Nursing is one of our newest programs, so I'm a little rusty, so give me some grace on that, but I can definitely help. Christina's amazing. She's our main advisor for that. But this is exactly how you get in touch with me and all of that. Any questions for me? No?
A PA program, PT
Yay,
program.
PT? Yes.
Public health program has multiple different degrees ranging from master's all the way up to PhD degree in public health. Pharmacy school, great pharmacy school. A lot of opportunities.
All right, thank you so very much. If you haven't signed in as well, go ahead, But thank you, everyone. If you'd like to stay and ask questions, you're free, but that is our talk. Thank you so much, Dr. Berg and Autumn. Thank you.
There you go. Also, there's some swag in here. If you guys want to take some on the way out, there's like lapel pins, koozies, they're for skinny cans, so like Celsius, Alani, those energy drinks.
Or another type of skinny can.
No, just energy drinks.
Oh,
Dr.
okay.
Berg. And I've got my cards as well.